In this video, we are going to talk about proteins. The first thing we are going to discuss is amino acids, which is the building block of proteins. Then we'll look at how amino acids would behave under acidic and basic conditions, and what would happen if it's placed in a neutral solution, in which case it will form zwitterion. Then we are going to discuss the primary, secondary, tertiary, and quaternary structures of proteins. And we'll also look at how we digest proteins, as well as doing some practice exam questions to solidify your understanding. So first, let's look at the roles of proteins. So proteins are present in both plants and animals. And proteins in animals have three functions. So the first function is to build new tissue and maintain those already present. So this is the key function of protein. The second role is to provide structural material for hair, skin, and muscle. The third purpose is to participate in biochemical reactions, such as biocatalysts. So it serves the same purpose as catalyst, but because proteins occurs in animals, so we call it biocatalyst. So another name for this biocatalyst is called enzyme. They play the same function as catalyst, which is to provide an alternative pathway for reactions to occur with lower activation energy. If you need more information about catalyst, refer to my rate of reaction video. Also, it can control entry or exit of chemicals. So this is called protein channel. It can also control communication between cells. So this is called hormones. It can also transport oxygen. This one you should be more familiar with. It's called hemoglobins. Also, it can protect bodies against disease. So this is antibodies. The last role of protein is to provide energy. But this only occurs in extreme circumstance. As we will discuss in later videos, the primary source of energy for humans is carbohydrates. And if we run out of carbohydrates, then the next in the line is fat. And if we consumed all the fat, the next in the line is proteins. Because proteins is the structural material for muscles, hair, and skin, and it has the crucial function of build new tissues and maintain existing tissues. When the body starts to use proteins to provide energies, this means you have not been eaten food for perhaps 1 or 2 weeks. Under normal circumstances, you will not be using proteins to produce energy, because by using proteins, you are essentially burning up your muscle and use that to produce energy for body to survive. So the fourth role is really not used under normal circumstances. The first three roles are the crucial roles that proteins provide to human body. So how is proteins made? We need to distinguish two things: amino acids and protein. So amino acids is the building block of protein. And amino acids looks like this. So you can find this on section 20 of your data book. So this is what amino acids looks like. We will discuss the structure of amino acids in detail in the next few minutes. But amino acids is not protein. It is one small part of protein. If you connect multiple amino acids together, you form proteins. So proteins is actually a polymer, and amino acids is a monomer. So poly means we have many monomers. So proteins means we have many amino acids joining together. So how is protein made? So it's made by something called a condensation reaction. So we already discussed the meaning of condensation reaction in organic chemistry. So it produces water. So by condensate, we mean that it produces H2O. So it's called a condensation, maybe I should add polymerization reaction between amino acids monomers. So this is how protein is made. It's made from amino acids. And amino acids is the building block of proteins. Don't get confused between these two things. So before we can understand protein, we need to understand amino acids. So we discuss amino acids first. So amino acids contains two key functional groups. So I will draw out a general structure of amino acids, and then we can look at what functional groups we have present as a revision for organic chemistry. If you need more information, please go to the organic chemistry videos. So the Z part is similarly to the rest of carbon chain. So remember I used something like this in the organic chemistry playlist. In here, the Z group is the side group, the side chain. And it can contain a lot of different things. Can be non-polar, can be polar, can have functional groups. So it is really a lot of variety of different Z groups. There are 20 amino acids, and there are therefore 20 different side chains. So maybe let's look at some real examples. So here, as you can see, each amino acids have different names. So you have AIA, you have ARG, you have CYS. So this corresponds to different side groups. So for alanine, so the AIA symbol one, the side group is CH3. For the arginine, you can see the side group is really long. And for ASN, asparagine, it's again a different side group. And as you could identify, the CONH2 is the amide functional group. And for aspartic acid, because it's acid, you can see we have a carboxyl functional group. And for cysteine, it is really interesting. So this contains SH, so it contains sulfur, and it would have interesting properties as we will discuss later. Glutamic acid, as you can see, although it's very similar to asparic acid, it contains one extra CH2 in the side group, and it still contains this carboxyl group. Glutamine is also very similar to asparagine with extra CH2 group. There are, of course, many more different types of side group. And in the exam, you are often asked to identify which amino acid it is from the side group. And if you are asked this kind of question, just compare what you have been given with what you have in the data book, and you should be able to find the correct answer. Just be very careful with this extra CH2 group, because often this is where the mistake will be made. So this is the side group. And another common characteristics among all amino acids is that we have a carbon at the central. And I think in the case except glycerin, where the side chain is actually just a hydrogen atom, the carbon at the central will always be chiral carbon. Because remember from the definition of chiral carbon is that the carbon is connected to 4 different things. And with the exception of glycerin, which is connected to 2 hydrogens, the central carbon is always connected to a different stuff. And so it is chiral. And now let's look at the other characteristic. So this is always connected to 1 hydrogen. Another feature is at this COOH. So the central carbon is always connected to a carboxyl group, as well as the amine functional group. So if we have a setup like this, it is called alpha amino acids. So it is a subgroup of amino acids. So amino acids is basically anything that contains amine functional group, which has the prefix amino, and also acid, which refers to the carboxyl group. So it is a carboxylic acid. And the alpha is a subgroup. So whenever you have the central carbon atom connected to 4 things, so the NH2 group, the COOH group, and the Z side group, as well as a hydrogen atom, it is classified as alpha amino acids. And this alpha refers to the central carbon atom, which is in most cases is chiral. So this refers to this carbon atom. Because the central carbon is chiral, then you know we have 2 different configurations. So actually we have this L amino acid and D amino acids. So those 2 are different amino acids, because when we have a chiral carbon atom, we can arrange things differently in space. And so they are just mirror image of each other. And the very interesting fact is, so almost all amino acids in plant and animals is L amino acid. So with a particular L configuration. And the reason is unknown. So why don't we get D amino acids? It's really something that we do not know, and something that scientists are investigating. So now let's move on to the side group. As I have already mentioned earlier, the side group determines the identity of amino acids. Because from the general structure, we know that all alpha amino acids contains the NH2 group, the COOH group, and 1 hydrogen atom. So what differentiates different types of amino acids is the Z side group. So if the Z side group is just another hydrogen atom, we know it is called glycine. And from the data book, you can see that we can have lots of different side groups, and each of them have a different name. Another characteristic of amino acids is the acid-base property. So let's have a look of the structure of amino acid once again. When you are drawing the amino acids, always draw in this configuration. So the bottom is always the Z group, and to the right is the hydrogen. Top right COOH, top left NH2. Because the carbon is always chiral with the exception of glycine. And if you draw it differently, maybe you are drawing actually a different molecule. So instead of drawing the L amino acids, you may be drawing the D amino acids, which would be incorrect, since we usually just deal with the L amino acids. The COOH, is acidic, so this part is acidic. And for the NH2 group, it is basic because it can accept one hydrogen or one proton, and the COOH can donate one proton. And this can have some interesting interactions. So when we have acidic environment, which is defined by pH of less than 7, amino acids behave like a base. So when it's acidic, amino acids becomes base. Why is this the case? On the acidic environment, we know we have lots of protons or H plus ions floating around. And because there are a lot more H plus ion than the amino acid, because it's have this basic property, it would accept one hydrogen plus ion. And this would mean when you accept H plus ion, this fits the definition of a base. And thus the amino acids behaves like a base because the NH2 group accepts a proton. And this is the definition of base. When something accepts proton, it is a base. And form the NH3 plus group. And this gives the molecule, the amino acids, a net positive charge. So let's draw out what happens with these amino acids under acidic condition. So the COH still stays the same because the environment is already acidic. We already have a lot of species donating their H plus ion. So there's really no chance for this COH, which you know is a weak acid, to donate its H plus ion. And we now have NH3 with a positive sign, indicating it has a positive charge. And another name of this is protonated. So accepted a proton, so become protonated. And on the basic environment, I think you can now predict the answer. The amino acids act like a base, like an acid. So why is this the case? So on the basic environment, a lot of things want to accept protons. And because we have this COOH group which can donate protons, then it will donate the proton. And when something donates the proton, it is considered as an acid under the definition of acid. So the COOH group donates a H plus ion and to form the COO minus group, giving it a net negative charge. So now the amino acid is negatively charged. So it will look something like this. So COOH becomes COO minus. So this means that the amino acids can behave as an acid and also as a base. So this is a really interesting property. Even more interesting is what happens on the neutral condition. So pH equals to 7. Then the acidic COOH would actually interact with the basic NH2 group on the same amino acid. And the result have a very funny name. It's called zwitterion, also called bipolar ion. The reason why it is called zwitterion is because it has a both positive and negative groups, but the overall charge is 0. So let's look at one example. So here you can see the proton of COOH is actually donated to the NH3 group. So one side is positive, another side is negative. And also, the functional group may also be affected. So let's have a look at one specific example to make things more concrete. For asparagine, as you can see, we have this NH2 group. So this means that under acidic condition, the NH2 would become NH3 plus. And this NH2 also becomes NH3 plus. So it would be double positively charged. For this aspartic acid, on the basic environment, both the COOH on the main chain and the COOH on the side chain would lose one hydrogen to become -2 charged. Similarly, for glutamic acid, we would also lose 2 hydrogens on the basic environment. So the side chain behaves the same way as the main chain. So this is the key thing you need to remember. And it is possible for us to form -2 charged or even -3 or +3 charged molecules if we have enough COOH or NH2 group. So let's do a practice question. Let's draw aspartic acid on the low pH and high pH. So let's first look at what aspartic acid looks like. So it has a CH2 and COOH on the side group. So let's have a look. So now instead of drawing Z, we need to actually draw out the full structure. So on the low pH, it means we have acidic condition. So it behaves like a base, except proton. So the NH2 becomes NH3 and we have this net positive charge. On the high pH, it is basic. We donate the H plus ion. And now we lose one hydrogen here as well as one hydrogen on the side chain. So both become negatively charged. So the overall charge becomes -2. So this kind of question is commonly asked in the exam. And I think it's very good to familiarize yourself with this topic and know how to draw different types of amino acid on the low and high pH. The general principle is on the low pH, it becomes a base. So NH2 becomes NH3. High pH becomes acid. COOH becomes COO. The next thing we need to know is something called essential and non-essential amino acids. So human proteins, remember proteins is basically a collection of different amino acids. So all the proteins in human body are made up by 20 amino acids. But we can only make 11 amino acids by ourselves. And the other 9 amino acids must be present in what we eat. So the other 9 amino acids that we cannot produce is called the essential. Those we can produce is the non-essential amino acids. It is non-essential because we can produce them ourselves. So it is really not an important consideration in our diet. And for the essential amino acids, because we cannot produce them ourselves, we must eat food to get them. And also, meat proteins, so like pork, beef, lamb, etc. They contain all 20 amino acids. So if you eat meat, then you should be all good. You can obtain all the amino acids you need. But for vegetarians, they need to be more careful. So they need to eat a variety of vegetables to obtain all the 20 amino acids. If they don't eat a variety of vegetables, then it is very possible for them to not obtain the essential amino acids, because vegetables does not contain all 20 amino acids at the same time. So now I think I have already introduced amino acids well enough, so we can continue on with our main topic of interest, which is proteins. So proteins is formed via this condensation reaction. How does this work? So let's start with two simple amino acids. By simple, I mean simple to draw, not necessarily a simple molecule, just to save us some time. So you can obtain this from your data book. We have 2H, so this is glycerin. So now let's draw a different amino acid with the side group CH3. So this is alanine. So here, how does these two monomers, or these two amino acids, react? We already covered this in organic chemistry. The COOH part will lose this OH. The NH2 group will lose one hydrogen. These two things combine to form water. And then we establish a link between the carbon atom and the nitrogen atom. So let's draw this again. So we have this CONH group, CH3, COOH. And this CONH group is called a peptide bond or amide link, because as you recall, CONH2 is the amide group. So CONH should have a similar name, so it's called amide link. And because water is a byproduct of this condensation reaction, it is called condensation reaction. And this process, of course, can be reversed via hydrolysis, which is defined as the addition of water. So if you add water, you can break this peptide bond or amide link into two amino acids. And every amide link that you form would result in one elimination of water molecule. And this thing is called dipeptide. Di means we have two monomers or two amino acids present in this molecule. And some of the naming conventions are: dipeptide means two monomers, tripeptide means three monomers linked together. So because you have three, you are expected to see, so this is 1, 2, 3. So you should have two peptide bond or two amide link. And during this formation process, you should emit or eliminate two water molecules. And polypeptide is many monomers, and proteins contains about more than 50 monomers. So as you can imagine, proteins is a really long chain of different amino acids. And in the exam, you can be presented with a small section of polymer or polypeptide chain. And you are required or expected to know how to identify the monomers that are formed, that contained in this polymer or in this protein. And the trick is to look at the side group. So by looking at the side group, you can easily identify which monomers it is, because the reaction only occurs between the two functional groups of the amino acids. So the side group remains untouched. And you can tell apart which is which by looking at the side group. So let's have a look of one example. This is a polypeptide with 4 monomers. From these two functional groups, you can see that it should be under neutral condition. So the COOH and the NH2 group actually reacted themselves and is formed the zwitterion. So overall we have 0 charge. So if you look at your data book, so firstly we know this hydrogen is always present in our monomers, so we just ignore them for the purpose of identifying which monomer is which. And then you can see the side groups. And the next thing you do is to go back to your data book. So maybe let's note them down. So we have the CH3, CH2SH, and this CH plus this benzene ring, and also CH, CH3, CH3. So with this information, you go back to the data book. So for CH3, you know it's adenine. And you can use the similar method of looking at the side group to identify the rest of the amino acids. So I have already put answers to this question here. Notice that I have used abbreviation, so PHEVAL, and those abbreviations correspond to the symbols section of the data book. So usually we do not write the full name of the amino acid, because sometimes it can be really long and tedious. So we use this abbreviation, and you can also do this in the exam. So this is how you identify amino acids from a protein or polypeptide. The next thing we are going to look at is the structure of the proteins. Remember that we are now talking about proteins or polypeptide instead of individual amino acids. So everything that comes next is related to proteins. So structure. So why do we study the structure of proteins? So the shape of proteins is important to their function. So the properties of proteins is really determined by the shape or the structure of proteins. And it can be divided into 4 different levels. The first level is called the primary structure. The primary structure is the order of the amino acids, which is the number, sequence of amino acids along the chain. So this is an example of the primary structure. So as you can see, we have different amino acids connected to together, and this is basically a polypeptide structure. You can see the CONH group, so the amide link. Because it's amide link, we know that it is joined by a strong covalent bond. And because it's covalent, it's difficult to destroy. The only way you can destroy this is to use hydrolysis, which requires a catalyst. For example, if you cook egg or you cook meat, the primary structure is unchanged. What is changing is the secondary and tertiary structure, which we will discuss later. So the primary structure is like the backbone of this polypeptide structure, and it's difficult to break down. So usually speaking, protein will remain a protein. So if you cook egg, you know it's still protein because the covalent bond is difficult to destroy. So that's the reason behind it. Of course, you can also use enzymes, which is biological catalyst, to destroy the primary structure. So it works the same way as catalyst. We will discuss the idea of enzymes in the later videos. So the next structure we need to look at is the secondary structure. Also, I should mention that the way that it is connected will alter the primary structure. So if you have something that looks like CYS with ARG, it is different from ARG with CYS. So if you have 3 different amino acids, you should have 3 times 2 times 1, which is 6 possibilities for them to combine different ways. If you have 20 amino acids, which is found in human body, is 20 times 19 times 18 times 1 possibilities. So there's really a huge variety of amino acids or proteins that can be formed by these amino acids. So the secondary structure is the attraction of one part of the protein chain to another part of the chain. So it gives a 3D structure: coiling, folding, pleating. So this attraction is the hydrogen bond between the CO group and the hydrogen of the NH group on another amide group further down the chain. So firstly, it is a hydrogen bond. So you know it is intermolecular interaction. So it's not covalent bond anymore. And because it is weaker, it is possible for us to disrupt the secondary structure of the protein. And when you cook egg, it is the secondary and tertiary structure that has been altered. And as you can see, that it is the hydrogen bond between different amide linkage, but it is along the same chain. So it's further down the chain. So it's the same protein polypeptide that are attracting each other because of this hydrogen bond. So this is one of the CONH bond. This is another of the CONH bond. And you can see that we can form this hydrogen bond between them. So this is called the alpha helix. So that's why it can coil together. So if you imagine a very long rope, and then each different sections of the rope are attracting each other, then it's possible for it to form this helix form. So very similar to DNA. So this is another one possibility. But remember, it is the hydrogen bond between the different peptide, between the different amide links along the same protein. So the distinction between the secondary and tertiary and quaternary structure is that the secondary structure is the same protein, and the quaternary structure is the different protein. So this is a very important distinction. We could also have something called the beta-plated sheet. Although they look like three different proteins, but in fact it is the same. And usually students make mistakes here and mistakenly think that this is somehow different proteins. And remember, it is not the case. It is still the same protein. It's just further along the chain, because proteins are made up by more than 50 monomers, so it can be very, very, very long. So next up is the tertiary structure. So it is the overall 3D shape resulting from interactions of side chains on the amino acids. So some possibilities are the non-polar side chains are hydrophobic. Because it is not polar, it does not attract water, so it's hydrophobic, afraid of water. Will fold inwards, so forms the interior of the molecule to avoid interaction with water. And similarly, the polar side chain will fold outwards because it wants to interact with water. The functional groups on side chain can interact with other side groups as well. And this can cause all different types of shapes and attractions of the overall protein molecules. So let's have a look at some of the common bonding that can occur between the side groups. So here, for serine, you can look up this on your data book. We can have this hydrogen bonding between the hydrogen and oxygen. So this is all interactions for the side chain. It's not the interaction between the amide linkage. The interaction between amide linkage is the secondary structure. Also, you can have ionic interaction. So this occurs when the NH2 and the COOH group react with themselves on the neutral condition. And this neutral condition means that the NH2 group gets a proton to become positively charged. The COOH group loses one proton to become negatively charged. And because they have this different charge, opposite attracts, so they can actually form a very strong ionic bonding between them. So this is a particularly interesting. So between two cysteine amino acids, remember the cysteine amino acids have a sulfur on the side group. And these two, if we have two cysteine side groups interacting together, they can actually form a covalent bond. And this will produce something called disulfide bridge, which is covalent bond. So when you have two cysteine amino acids bonded together on the side chain, the tertiary structure becomes really, really strong. And it's very difficult to disrupt. As strong as the primary structure, because we have this covalent bonding. So it is the strongest tertiary bond, and it will be the last bond to break. We can also have dispersion force. So that's for the two non-polar side chains. And it is very weak, as you would expect. Of course, because we can have hydrogen bond, we can also have dipole-dipole interaction, since they are essentially the same stuff. The next structure is called quaternary. So this is the overall shape, resulted from several peptide chains joining together to form overall structure. Also can include non-protein molecules. So it is usually held together by dispersion force. But hydrogen bonding, ionic bonding, still possible. So essentially you have different proteins joining together. So that's the quaternary structure. Protein 1 joining with protein 2, and then they joining with protein 3. So because proteins themselves are very complicated, with primary, secondary, tertiary structures, when you are joining different proteins, it becomes even more complicated. So that's just something you need to know. Usually not tested in the exam, but you need to know the differences between the different structures. So primary, remember, is the arrangement of the amino acids. Secondary is the attraction between the peptide or the amino amide links. Tertiary is the interaction between the side chains. Quaternary is the interaction between different protein molecules. So very important distinctions here. The next thing we're going to look at is digestion of proteins. So the body does not store amino acids. So we need a balanced intake of amino acids every day. And proteins is converted to glucose or fat when it's digested. And because proteins contains amino acids contains nitrogen. And nitrogen is a very special element. So nitrogen atoms converted to ammonia. Ammonia is toxic at high dose, and which is then converted to urea. So a very good fertilizer. And let's look at what urea looks like. So a very beautiful shape. As you can see, it's very, very symmetrical. So doesn't have a lot of hydrogen environments. So just recap, should only have one hydrogen environment because of this symmetrical shape. So it is polar. It is very soluble because oxygen is electronegative, nitrogen is electronegative. So you have really a lot of potential to form hydrogen bond with water. So it's polar, it's soluble, it's not toxic. So low toxicity. Because it's soluble, we can eliminate in urine. So I guess urine and urea are very similar name because they usually go together. So that's just what you need to know about digestion of proteins and this urea molecule. So it's written like this, CONH2. So when you see this in the exam, it is urea. So let's have a look of some practice questions. So a two multiple choice questions that is very common in the exam. So this is from the 2017 VCA exam. This is from the 2018 VCA exam. So which of the following represents zwitterion? So both positive and negative charge. So let's have a look. So B only have positive charge, so not possible. And A only have negative, so not possible. So now let's look at option C and option D. Option C, you can see, have an overall -1 charge. And this is not the definition of the zwitterion. The zwitterion is overall neutral. And as you can see, D is overall neutral. So D is the correct answer. And this question asks us which is a dipeptide. So 2 amino acids. Alpha amino acids, remember, has the general shape. It looks like this. So all of them contains this peptide link. So which is good, because for a dipeptide we need one peptide link or amide linkage. So for number A, it doesn't look like alpha amino acids. So for this amide amino acids, it is alpha amino, but for this particular one, it doesn't look like amino acids. So because amino acids, we should see NH2 directly connected to this carbon. So so A is incorrect. Remember, the NH2 should be connected to the central carbon. And here the central carbon is connected to one carbon, another carbon, and 2 hydrogens. So definitely not alpha amino acids. So it is another type of amino acids, but not alpha amino acids. For B, is similar. So the H2N is too far away. So it should be here to be alpha amino acids. So B is also incorrect. Let's look at option C. So C should be good. If you look at these two central carbon atoms, so it is connected to hydrogen, the COOH group, another hydrogen, and the side chain. For this carbon, connected to COOH group, side chain, another hydrogen, and H2N, nitrogen. So C should be correct. And D is the similar problem. This side is okay. So this is the side chain, this is hydrogen connected to another COOH group connected to the H2N group. But for this one, you can see that the C here should be directly connected to COOH in order for it to be alpha amino acid. So we have this extra CH2, which makes it incorrect. So C is the correct answer. Also, I should just mention, forgot to mention this. When you digest proteins, you are essentially doing a combustion reaction. So you will be producing urea. Because you are doing combustion to release energy, you are also producing H2O and CO2. So that's the two other products that can be formed and will be formed if you digest proteins. And of course, this is a really undesirable circumstance because we don't want to digest proteins. Proteins are used as a building block of our muscle, our skin, our hair, and it is really the last resort that the body turns to because it doesn't have sufficient energy, is to digest proteins. And the end product is the same as digestion of carbohydrates, water and carbon dioxide.